Welcome to the PharmaSource podcast. Today's interview is a conversation with Alexander Aust, a consultant specialising in the manufacturing of lipid nanoparticles. LNPs are a key component of mRNA vaccines, and as such saw a huge acceleration during the COVID-19 pandemic. In this conversation, Alex explains some of the innovations in lipid nanoparticles and the challenges around their manufacturing. As a start then, could you give us a bit of an overview of what lipid nanoparticles are, LNPs, and their primary uses in medicine, just to get us started?
Sure. So lipid nanoparticles, or LNPs, are one of two classes of drug delivery vehicle, basically like an Uber for the active drug substance to get somewhere in the body. So you have the viral and the non-viral. So viral drug delivery methods are like AAV, adeno-associated viruses. And then, non-viral are like lipid nanoparticles, liposomes, polymeric nanoparticles. And there's there's several differences between those classes. Viral drug delivery is most prevalent with FDA-approved drugs, but LNPs are coming for them. Viral is typically may have more toxicity. Usually, though, that you do get a pretty high level of transfection, if you're doing mRNA or gene therapies. And then there is the drawback, of course, that viral manufacturing is more expensive. So lipid nanoparticles is the synthetic biology where you're going through and you're using these different lipid building blocks to come in and circle up and encapsulate, basically completely surround from the outside, either an mRNA like in the COVID vaccines or these different types of, small molecules, to be able to get it to somewhere in the body. So the liver, if you need something, go to the lungs, then you're able to basically take a drug that was never be able to get to where it needs to go. But if it could just get there, it could work. And LNPs are functional in that way that they help the actual active drug substance work. So before these types of drugs, it was really a lot of focus on the drug substance or the API itself. But with LNPs, it's a mixed combination. And that's what makes it so complex.
And the market really exploded during COVID-19. Why did LNPs become such an important component when it came to the development of mRNA vaccines through the pandemic and beyond?
Yeah, that's a really good question. I think it's partly because of just the physiochemical nature of, again, the mRNA, which was the active drug substance. So the mRNA is what ultimately gave everybody with COVID that ability to be, develop the immunity. But mRNA is this physiochemically negatively charged thing. So one of the main building blocks of the lipid nanoparticle, one of the lipids, and traditionally there's four, is a cationic lipid. And what that is cationic is just a positively charged lipid. And what that does is that, when mixed, allows the lipid nanoparticle to completely form a lipid layer around the actual RNA and then form another on the outside. So it really lent itself well to that. And, mRNA and LNPs, or in one derivative of it, or in some derivative of it, has been done for decades, actually. So over the course of time, so before COVID vaccines, the only other approved actual LNP drug product was Alnylam's Onpattro. And I think it got approved in August of 2018. It wasn't an mRNA, but it was an mRNA technology. So it was small interfering RNA. So still, again, negatively charged RNA gets encapsulated by these lipids. I actually, a couple of weeks ago, was speaking with Dr. David Curiel of Washington University. And back in 1995, he published a paper, I think it was characterization of a messenger RNA polynucleotide vector or vaccine vector, something like that, which is a long way of saying, even back in 1995, we were looking at these non-viral drug delivery methods for getting mRNA and allowing that to get into the body. So over time, the liposomal drugs, which was the forebearer, I guess, to the lipid nanoparticle, and these suspensions and emulsions, these were approved drug products starting in the 90s and up till now. But really, around the time that CRISPR-Cas9 really started to catch on uh, after the Nobel Prize was awarded, and gene editing and gene therapy started becoming something that could actually be taken out of academia and into industry. Well, there was actually already this drug delivery vehicle that was kind of waiting for it. It was just, lipid nanoparticles were in a way just waiting for a worthy cargo to take and be able to go through. So then it
Hmm.
came down to scaling up manufacturing, targeting, and of course, dialing it in for what COVID-19's purposes were, which were immunity for the globe.
Thank you. Very interesting. And I'm interested, I think, to know, Alex, about your own journey as well. So, how did you end up in this particular field?
Well, I think in this particular field, I guess full time is back in 2012. I started working at a company called Sigma-Tau Pharmaceuticals, and it was a company. So we owned several approved drugs. They were actually orphan drugs, which means they were for very small patient populations. And one of those drugs, so the site I worked at was the site that the liposome company built to manufacture the approved product Abelset. So it got approved in 95, and I come along in 2012, and that's a commercial liposomal drug product, and those are rare. I think at the time it was maybe there was 10 to 13 of those. So my exposure to these non-viral delivery vehicles at a commercial level was
Mhm.
kind of, I guess, is different than a lot of people because those sites just didn't exist and the demand was there because again, it's orphan drugs. There's a very small network. There's a very small group of people working in the field. And really, Abelset couldn't get more complex. It's a 7-day closed, so aseptic manufacturing process, which means you can't have exposure to the outside. And you have to sterile filter everything in, and then you push it around these different tanks and mills and things for like 7 days. And then you pipe it directly into your fill finish suite. So again, all of these drugs up to date are parenteral, so they have to be sterile and in a vial.
Mhm.
And then you filled that. So we also manufactured these other drugs that we had were pegylated proteins, though. So PEG, it's polyethylene glycol, is a polymer. As it functions to LNPs, that's another building block. So you get these, so you had that cationic lipid that wraps around the RNA. Well, this PEG lipid, can function for stability, and it also can function for a stealthing effect in your body. And that's what it was kind of doing with these proteins. So we'd conjugate the PEG and the protein and put together. So that was a drug product. So that was like 2012 through 2016. I think 2016. So, that was commercial products. And of course, we were doing the later stage stuff as things were coming along. There was a liposomal drug in 2016 that we were taking from phase 2 and 3, and we had done the submission, but unfortunately, it didn't meet its endpoint. It was a nebulized drug. So what that means is this was a liposome that was going to go into a nebulizer, aerosolized, and go to treat the lungs. Unfortunately,
Mhm.
it didn't work. Um, Kind of how I got to now was so over 20, in 2017, Sigma-Tau sold off all its assets, and we were still manufacturing those products, but we now existed as a CDMO. So, as a CDMO, we needed to rebrand and launch and take on these new products to, survive and grow. And we became Exelead. And because I had by that point worked in a lot of the different departments associated with taking these technologies to the clinic or into GMP manufacturing so that they could go into patients. I became the head of business development there, and from 2017 to 2020, that's when we really started reaching back into the preclinical and clinical projects. So that's when also not only the earlier stage stuff, but that's when I really became intertwined in the drug substance development and manufacturing and supply chain as well.
Mhm.
Because as we found over time, the mRNA or these oligonucleotides, their interaction and efficacy is heavily dependent upon the lipid nanoparticle. And so kind of co-developing them as along the way is of greater and greater importance the more we find. So really that's when I started getting those knowledges around the knowledge around the mRNA and how complex not only its process was at the time, but for, let's say, individualized, personalized cancer vaccines that we were manufacturing in 2017, 2018, but also being able to make sure that the finished drug product was sterile released, all the quality methods that we were sometimes developing for the first time because it's going for first in human drugs, right?
Mm.
So there's no precedence for it. So that's when you get like those first generation mRNA and gene editing companies, I think. So that's when you had like, the Intellias and everybody started going into the clinic and we were manufacturing and really honing in what those critical quality attributes were and how would we control this manufacturing process. So I left there in February of 2020 to start consulting for drug delivery systems and parenterals full time. And then in March 2020, COVID hit. And really the focus has become mostly on LNPs.
Very much feels like being in the right place at the right time to be an LNP specialist just as COVID's taking over.
Absolutely. That timing allowed me to work. I'm very thankful for that because not only the experience, but that timing allowed me to help a lot of different programs at
Mm.
once and really optimize my efficiency with trying to do COVID related things as well as still getting these other products into the clinic.
Where you sit now, then, Alex, in a consulting role, working across a number of different customers, what do you see as being the main challenges that people are facing when they're manufacturing LNPs, particularly trying to bring them up to scale? What are the common challenges that you keep coming up against?
When we have decided on what the target is in the body, the cell, or the tissue, what the therapeutic needs to do from an indication perspective, you have to screen different lipids for that purpose and conduct these different tests and be able to manufacture and make a lot go a long way. A lot of
Mm.
sample go a long way, or a small amount of sample go a long way. And really, what it was initially was actually, COVID came around and we scaled that out more so up the manufacturing processes. But when you look at it, there was actually this void of having any applicable technology that could get down to the really small levels. Now there was a Precision Nanosystems, now Cytiva, was really the only player that did microfluidic mixing or these types of technologies that could do like micro or small milliliter samples so that you could see what is the best formulation of LNP that works the best. How, I'll take the mRNA, I'll encapsulate it in a few of these different, you know, formulations, try these a few cationic lipids. Which one got to the liver best? Which one got to the lungs best? Well, so we shot kind of down as an industry trying to get to this race to the bottom. In response to COVID vaccine production, like those global levels, like I said, instead of like scaling up, it was more like scaled out. So the real difference between scaling up, scaling up, you're taking this existing resource and making it larger. So this pump, and it all comes down to these pumps and the actual microfluidic mixing device, either a T junction, a lot of times Y junction, or some kind of in-pigment jet mixer, where effectively how you make an LNP is you take the lipids from one side and the mRNA from this side, and they're both in their buffers, and they come together and they mix turbulently and encapsulated because, again, of physiochemistries and your formulation optimization, controlling these flow rates. Now, the issue really was that instead of making this a bigger process, we really just added more mixers in line. So
Mm.
we basically just said, hey, it's just like a car engine. Instead of a V6, now we had a V8. And it's just a different thing. So the other thing that I think is really interesting from the perspective of seeing this space grow and being heavily involved in the clinical operations before COVID was to now, it's really great how there's additional analytical resources and different types of technologies out there. There's more mixers and more abilities to formulate these LNPs than ever before. There's a lot of problems with, handling in some organizations, there's a lot of problems handling the large amounts of organic solvents that it takes to actually produce these LNPs. Organic solvents being, you don't think about it, but when you make an LNP, you're either using like a lot of ethanol, like 200 proof, which is highly flammable and explosive. So you have to be able to make sure that, when we're in the GMP settings, we've scaled up, we've protected everybody, we ground the room and the equipment and make sure we're ventilating so that people don't go past their acceptable daily exposure limits, right? So you also have to do downstream processing, and the tools that have become more available and more the shared information that's been nice is, if we're using hollow fibers, filters or cartridges, or however we're going about doing the scale up to the GMP filtration process to take these organic solvents out, there's more information about the selection of those types of components than ever before. And also one of the difficulties is that, I had mentioned that this has to be sterile, like ultimately when it goes into the patient. So, your drug substance can come into a manufacturing site and then you encapsulate it. Well, you can't irradiate it. You can't cook it at a really high temperature or you denature everything. And so what you have to do is ultimately pass it through the terminal sterilization process is you pass it through a filter, like a 0.2 micron filter. And the selection of that cartridge and the materials and everything, that's gotten better over time. There's just more options. But a really big issue still remains that when you transition most from preclinical, clinical, these smaller, to preclinical, small scale, bench scale animal models to large scale, you have to generally go from that microfluidic or smaller mixing process that I described, and you have to do a process development to now go to this GMP batch scale, or now there's continuous manufacturing models. But really what you have to do is you have to switch the mixing technology. So you have to go from microfluidic to something more in line with like solvent injection. And so what does that mean? That costs time, that costs money. And you have to make sure that when you've changed the manufacturing process, you haven't changed the drug itself either. You haven't changed the nanoparticle. You haven't changed how much of it gets in, how much of the mRNA gets into the LNP. It has to be the same. So, if you run your tox batches, for preclinical at a really at these smaller manufacturing scales, you'll have to show to the regulatory authorities that, hey, we've changed it now that we're dosing patients, but it doesn't have that impact. Now, when I work on these projects from the regulatory side, we can plan ahead and we can look and say, hey, well, we'll use this material from this batch to satisfy these needs, and there's strategy around that.
Are there any particular regulatory requirements from the FDA around LNPs which are front of mind when you're trying to scale up these processes?
Yeah, I think applicable ones that people can look at. So to answer your question straight at face value is no, there's no guidance directly from the industry like the FDA guidance documents. In 2016, because again, back in 2016, we were working on liposome products, and that was the only thing that was up to that point that was actually in the clinic or commercially approved. So they actually released a liposomal, product manufacturing guidance, which has some good tidbits in there as it relates. But again, that's like 6 years old and outdated in a lot of regards, or was just missing information. But that type of guidance document existed really for only orphan-based drugs. It's what's really incredible about the technology is it can go from an individual patient dosage or an orphan drug, so it's like less than 100,000 people in the US annually, to everybody in the world. So that's a very versatile technology. In that way, recently the FDA came out with a draft guidance for platform technologies. And these platform technologies meaning what do you need to bring on a regular regulatory basis. So I use this LNP that's proven to deliver this mRNA. I swap out the mRNA, the LNP is the same. What do I need to show from this LNP to satisfy the FDA? They've provided those types of things. Those are actually in draft guidance. So receiving comments back now.
Thank you. And you mentioned continuous manufacturing as well. How much of an impact has that made so far in LNPs?
I would say as of right now, not much. It's in its infancy as it was related to other technology. And you really kind of got to nail down what continuous manufacturing truly means in a lot of these applications.
Mhm.
Sometimes, again, it's marketing is a wonderful thing, but I often ask when I'm looking at these different companies, these new technologies, and analyzing it, I'm trying to find out what's innovative and what's different. And in a lot of the ways up until this point, batch manufacturing versus continuous manufacturing is what people often think of, that you're doing like this campaign. So at a larger scale, continuous manufacturing means you never have to come down. You have a reduction of waste and things like that in the input materials. Well, as you're processing that, that sounds theoretically to me very good at satisfying the need for meeting the quick demand of the next global crisis. Whenever, a pandemic hits again, would be able to manufacture that at larger volumes. I think also it was satisfied through batch manufacturing as well, because what actually is surprising is, from my observation, one of the rate limiting steps wasn't actually finding enough places or equipment to manufacture the LNP. It was from taking that LNP in that finished bulk drug product form to making it a finished drug product form by filling it.
Mm.
So you have to filter it. So there were fewer sites that were capable of doing the massive. So you've generated a lot of volume of bulk product. Now you have to get enough thousands and thousands of vials, a very small fill volume. So that required a lot of precision. The other aspect of the continuous manufacturing process is how much of the downstream processes are you putting in place? So if you mean continuous manufacturing in the way that the unit operations, from setting up the equipment to buffers and putting everything in the system, and then you encapsulate, then you do downstream filtration like we had talked about, and being able to analyze that along the way has value as well. These process analytical technologies, PATs, that the people are putting in these systems are great. However, again, is when you're doing these process development activities, you need to be careful about what data you're collecting, because if you collect a bunch of data and you submit it to the FDA, you have to describe it, you have to answer for it. And if early on in the process, there is an anomaly or something that you can't explain, it may give you hiccups. Now, I'm always in favor of more data and better
Mhm.
process and quality characterization. So I'm not really afraid of that. But again, as we had previously mentioned, the regulatory guidance isn't there on LNPs, much less this continuous batch manufacturing technology. So being
Mm.
able to implement that at these different places has a certain amount of regulatory complexity as well. And I think these have been referenced to be great for point of use, or being able to, manufacture them all in a shipping container, getting your mRNA and your LNP all together. But I think getting the drug substance, getting your lipids manufactured, getting your drug substance manufactured, getting your drug product bulk manufactured through the LNP, and getting your finished drug product is four very different manufacturing steps.
Mm.
So there are companies like Quantum Biosciences that are trying to take their continuous mRNA manufacturing system and plug that straight into an LNP system that they're developing. So these processes are happening, but then the adoption of that, mostly up until this point, LNPs outside of COVID and liposomes and these non-viral delivery vehicles, like even PNPs, polymeric nanoparticles, they're for rare diseases, they're for orphan drugs. You may have, like I said, one patient, or you may have, okay, we're only going to need to make a batch to satisfy this first study or this first part. Maybe you're at the tens of liters of scale. So it's
Mm.
not, the demand may not be there. Having this in the wings, having this be able to, again, so now you've done continuous manufacturing, but can you do continuous manufacturing at the smaller scales?
Yep.
Can you get down lower? With the characterization work being done at the early phases, again, what's the difference between switching from microfluidic to this continuous manufacturing system? Now, actually, you don't actually have to switch anymore. There are microfluidic systems now that are GMP scale that you would just change out the consumable mixer each time. And now, so you wouldn't necessarily have to change it for these rare disease drugs again. But again, there's going to be a cost associated with these more complex consumables. So
Mm-hmm.
always a balancing act, and I think continuous manufacturing has a place to play, but I'm not exactly sure where that is right now.
Taking a step back from the manufacturing processes, how do you see the field of LNPs evolving over the next decade or so? Do you see them moving out of vaccines and gene therapies into other areas?
Yeah.
What are those applications which are on the horizon?
Yeah, I think even just from a fundamental standpoint, I had mentioned that, there's the standard building blocks of LNPs. So you'd have that cationic lipid, you'd have that PEG lipid, and some helpers like phospholipids. And as the technology advances in polymer and lipids and material sciences, it's giving us more access to more building blocks. And so we can make
Mm,
better and better LNPs. For instance, Cytodime, they've got this platform that actually uses gangliosides instead of a PEG lipid as both a PEG replacement and a targeting lipid. So that really changes the way that, we look at the function of cationic lipid. So I don't even need a cationic lipid anymore to be functional and target and things like that's taken out elsewhere. There's also the ability to develop these different drugs that I had mentioned in 2016. I was working on a liposomal drug that was for nebulization. I wonder if that failed partly because the liposome would, I don't know this, and I just always wondered, when you nebulize it, it's a shear process. There's forces. Were we disrupting and destroying those upon the delivery? Well,
mm.
we know that can be a problem now in 2024. So these different companies are using better LNPs and better building blocks to survive nebulization and the shear forces and temperatures and different things like that. So you get more stable nanoparticles to get delivered. So I think you're getting different delivery methods as well. So, you're not getting an IV like you are getting for Onpattro, Alnylam's LNP. You're getting shots on goal for nebulized drugs, which is great for the delivery to the lungs. There's also the aspect of the active targeting. So active versus passive, simply put, passive just means there's nothing really, the lipid selection itself has allowed it to go to, and control of the particle size allows it to go to the liver. A lot of everything goes,
Mhm.
blood goes through the liver for filtering. And so for the mRNA vaccine, that's where, the target is. Well, now how do we get to target even harder to reach areas than the liver? And that's through this active targeting. So the LNPs themselves not only are getting more complex because we have the ability to use different building blocks, but now we can decorate the outside of them. Right. So
Mm.
now we're thinking, okay, well, we got this nanoparticle that gives us the best chances of getting into the cell, gets us, because it has to target, it has to get there, then it has to release its payload inside in the cytoplasm. So different bioconjugation methods have been used to affix different proteins, so like antibodies, to target on the outside of these LNPs to get into these regions. And then again, there's no reason that it is just restricted to mRNA. mRNA just gets talked about a lot because of the COVID vaccines and because LNPs are almost exclusively used for mRNA delivery these days, because there's both the proven clinical track record, regulatory precedents, but there's all those different like pDNA, plasmid DNA. There's small molecules, there's peptides. You can encapsulate a lot of different stuff, and I really do think too the LNP is just one class of these lipid-based nanocarriers. There's liposomes, there's lipoplexes, there's suspensions. There's different ways that we can configure this. But
Mm.
it's really a lipid-based nanoparticle is really then everything else is the nuanced difference of what those actually look like.
Where would you see the market going over the next 5 to 10 years or so? I mean, are there new therapeutic areas that it could be used in?
Yeah. Yeah, so cell and immune therapy with CAR T and everything else like that, has not even really been tapped yet. So being able to
Mm.
target the immune cells in immunotherapies is going to be huge. Um, a lot of these generation two, I'll say, so there was all of the biopharma companies that existed before COVID, and then now there's all the companies that existed from COVID to basically 2023.
Hmm.
They're all now getting, that second generation group's getting ready to go into the clinic or it has just started into the clinic. And we're getting to see those results in that feedback for efficacy and further proof that, hey, we can use this to treat cancer. Now, cancer on a wide scale, impacts millions of people. But I had also talked about personalized cancer vaccines. I think those are
Hmm.
absolutely going to be a thing once all of the supply chain issues are satisfied. We'll be able to take some sample from someone and then we'll be able to take that tissue, sequence it, get your RNA, get it encapsulated, get it filled, get it back to the patient in less than 2 weeks,
Hmm.
then you're really moving on a lot of these rare disease drugs again. I think also there's huge ways to grow outside of just cancer
Hmm.
vaccines and vaccines as well. There's small molecules and different things like that and reformulations as well. I think you can look at improving upon what has existed now for 30 years and say, well, maybe there is just a better way. Maybe if we can keep that in this, maybe we can keep that same drug substance in the bloodstream for a little bit longer, then we can decrease the dosing and things like that.
Hmm.
I really see the gene therapy and gene editing market only ever growing. I
Yeah,
mean,
yeah.
the gene editing is going to be a big thing, with the CRISPR-Cas9 and those companies that are really taking that forward, being able to treat those types of diseases that there's just no other option is really never going to slow down.
And finally, just interested in what advice you could share, Alex, with people or companies who are striving to innovate within the field of LNPs. What advice, what best practices would you like to share with people who are trying to make an impact in this field?
Yeah, if you're trying to innovate, really there's a lot of room still.
Mm.
Now, I think what a lot of people, when they became more aware of lipid nanoparticles, they maybe thought the field was a little bit more seasoned or older and developed than it is. It's not by any means a mature technology. So
Mhm.
yes, the liposomes have been around for a while, but again, the mRNA, LNP, those interactions have been around in the clinical setting for a short period of time. So, and a lot of what you're going to need to do is generate data. A lot
Mhm.
of people, I hear a lot of really good ideas, but in this industry anymore, it's 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 not saturated in the way of data, but it's saturated in the way of, I think, of just good ideas. And
Mm.
that's what's going to hold people back in funding too. So being able to, I would say there's ways of doing that and taking advantage. The NIH and these different groups have ways of getting non-diluted funding and things like that. And a lot of it's related to LNPs, delivery vehicles, both on the manufacturing equipment to improved nanoparticle formulations. Everybody wants to get across the blood-brain barrier, the BBB.
Mm.
Everybody, if you can take, and people are trying to get LNPs and liposomes to do that. So if you can get that across there, that's a huge. Again, now you've brought the brain as an area of an organ that's a very difficult to treat.
Mm.
The other thing I would say is the industry infrastructure, like through CRDMOs, has been established pretty well now after COVID. I would
Mhm.
say in that area, there's a lot of options, no matter what market you're in, the US, EU, or Asia, depending upon what you need. There's a lot of people out there that can help if you want to outsource some of those activities to save money and get some expert opinion too. I think as far as technologies go, again, there's a lot of new analytical technologies out there that were not available or were just not even thought of to be applied to LNPs prior to COVID or even now. It's not a requirement through the FDA, but I think it's a great idea to get cryo-EM data, which are these basically these pictures you get of your lipid nanoparticles to see, okay, a lot of the time we're going for encapsulation efficiency, how much that mRNA got in there. Well, how much of that mRNA, so that's a number, right? That's a percentage. Well, what does it look like in there? Is there some that's on the outside too? So there's these ways that, the FDA, while not requiring it for phase 1 submissions, I think actually the only regulatory agency is in China that requires cryo-EM. They're a little stricter on physical data for the IND, their version of an IND submission for phase 1. But it's really great, I think, to get these pictures earlier on in the development process so you actually know what your product's doing. So again, that wasn't really on the table back in 2016. We didn't really think about it that way. But now it's, hey, if we're doing this formulation development, let's make sure it's behaving like we think it is. And again, if you're able to defend whatever your decision is with scientific justification and it's quality product sound, the FDA will go for it and talk you through it and coach you. So they have their times to talk with you before, the first meeting and submissions and everything else like that. So if you have any questions, most of the questions, again, are around impurities. I tell people not to get too worried about it because you have to generate data and you have to figure out your drug product's process, your drug substance and its process. And if you don't understand those things, you're not going to understand those things necessarily up front. So just keeping in mind not to set your specifications too tight in the beginning, because you can always tighten them. The FDA doesn't like it when you try to back them off. So from a regulatory perspective, give yourself as much grace in the development process as you can by not painting yourself into a corner. And just, ultimately, innovation around getting outside extrahepatic drug delivery is going to be welcome. And I don't think it's time wasted because I only see this growing in the future.
Alex, thank you so much for your time.
Thank you so much. Thanks for having me. It was great.
Experience the future of contract manufacturing at CDMO Live 2025. Join us at the World Trade Centre Rotterdam on May the 7th and 8th, 2025 for an exclusive event that brings together the sharpest minds in biopharma outsourcing. For 2 days filled with market insights, best practices, and partnering. Early bird tickets are now available, so head to cdmolive.com to download the agenda and to book your ticket.